Hola, welcome to my channel. ¿Cómo estás? How are you? Muy bien, I'm also fine. Today we are going to dive deep into the structure of DNA. So buckle your seatbelt. Please grab your pen or pencil and a piece of paper and follow closely till the end of this video so that you will be able to perfectly draw and understand the structure of DNA. As last we talked about the definition of DNA, we also looked at the difference between DNA and its cousin. We also looked at the shape, which is leather-like but helical in shape. We also looked at the nuclear bases. So today we will continue from there. For the nuclear bases, they are grouped into two. We have the purines and pyrimidines. The purines are the adenine and the guanine, and the pyrimidines are the rest. So the cytosine, thymine, uracil. Either if it is a DNA, it is thymine. If it is an RNA, it is a uracil. So those three are the pyrimidines, and the two adenine and guanine are the purines. So DNA is a nucleic acid, and nucleic acids contain polymers of nucleotides, and the nucleotide contain three main parts. The first one is the pentose or 5-carbon sugars. The second one is the nitrogenous bases, and the third one is the phosphate backbone or phosphate group. Yes, so buckle your seatbelt, and I promise to make it as simple as you can understand. So if you don't have a pen or paper available right now, you can save the video and later on play slowly or pause, and you will try to get your structure down to know about it and then for future reference. So pentose sugar, pentose because it is a 5-sided figure. So as we have pentagon to be a 5-sided figure, as you all know, a pentagon, if you are drawing a pentagon, we have at the apex a vertex there. When you come down another vertex, you go this way, another vertex, you go up another vertex, and you close another vertex. So 5-sided. So that is how we are going to draw the nucleic acid or the DNA. So the first one is O at the apex, at the top, O. So from O we draw a short line to C. That C is the first carbon, and on the first carbon we have the nitrogenous base, so N base, and then H. So you draw a C, and then you draw a short line. On top you do N base or the nitrogenous base, and below you do the H. H is the hydrogen element there. So with the carbon 1, that is where the nitrogenous base is located, and the nitrogenous base can be any of the 4 bases we've mentioned earlier. The A, T, C, or G can be any one of them. And from the carbon 1 you go to carbon 2, and on carbon 2 we have H, and below we have OH. Now pay attention, this is very crucial and critical. I want you to get this very well. That the OH, because there is presence of oxygen, and if there is a presence of oxygen in this structure, then this structure will be called a ribose sugar. Okay, but as we've mentioned earlier, or I've said earlier in my previous video, that DNA is deoxyribose sugar or nucleic acid. Deoxyribonucleic acid. So meaning it has lost an oxygen, and this is the point. This is the carbon 2, on carbon 2, that it has lost that oxygen. So you have HH, not HOH. No. So H on top, then when you come down, another H, meaning you draw a line on carbon 2. You draw a line. So you have those who have done what do you call it, chemistry, you understand perfectly what I'm talking about. then below you have HH. So that will tell you that this has lost an oxygen, and a structure that has lost an oxygen like this will be called a deoxyribose sugar. But if the OH is present, it's a ribose sugar. So from carbon 2, you make a curve to carbon 3. Now also pay attention, this is very crucial, you must get this one very right. On carbon 3 we have H and down OH. Mind you, this OH is not the same as on the carbon 2 OH. This OH will help us to join what do you call it, nucleotides together. So as I've told you, on carbon 1 we have nitrogenous bases. So this nitrogenous base can be A. So for instance, the gene that codes for the I is AATGC. So AATGC, it has to join together. So we cannot have only A to code for the I. No, we have to to get a gene, and that gene, the full gene is AATGC. Sorry. So A would have to join to A, and that A, the second A has to join with T, and that T has to join with G, and that G has to join with C. So this is the point on carbon 3 that the joining will occur. So if there is OH present, meaning it is called a sticky end. So meaning a nucleotide can be added, more nucleotides can be added till we get to H. So if it is HH on carbon 3, meaning it's a blunt end, meaning no other nucleotide can be added, meaning the end of that particular gene. So that is how genes are being placed, or instructions are being placed. So they are connected to each other till they cannot connect anymore, and this is what will cause that connection to occur or not to occur. So if we have a gene that codes for the I, and let's say that it is AATGC, then at the end of C you have HH. On carbon 3 we will not have OH, but at the end of the first nucleotide A, at the carbon 3 we have OH. Second A we will have OH. The T we have OH, G we have OH, but on C we have HH. So this is where I want you to get very clear. So this carbon 3, the OH on carbon 3 is not the same as the OH on carbon 2. So we move to carbon 4. On carbon 4 we have HH, and carbon 4 bridges or closes the structure, and it moves back to O. So isn't this interesting? Because we said the pentagon is made of 5 parts, right? But then we have gotten the 5 vertices, but there is one carbon missing. But we said this is a 5 carbon sugar, right? The nucleic acid or the DNA is a 5 carbon sugar, but we've gotten only 4 carbons. Where is the one? If you know where the one is, kindly put it in the comment section below. But I'll tell you, this is interesting. As the carbon 4 closes the ring and makes the ring complete, carbon 5 is very crucial, and it hangs outside the structure, causing or giving us the attachment for the phosphate, as we've mentioned the 3 parts. So this is where the phosphate backbone comes in. This is the carbon that connects to the phosphate backbone. So from carbon 4 we have a line. You draw a line outside, going outside. So the carbon 5 hangs outside the structure, and it serves as the attachment point for the phosphate group or the phosphate backbone. So usually you get it abbreviated as O-CH2. Okay, so on the carbon 4. So that is the structure of the nucleotide or the DNA. I hope you've gotten it, and I've tried my best to make it as simple as that. And if you haven't gotten it, play it over again and pause to take your notes, and at the end I promise you get it vividly as it is. It's very simple. Just take your time and follow the step by step. So let me take it all over again. We have the O at the apex. From O we go to carbon 1. On carbon 1, nitrogenous base and H. From carbon 2 we go to carbon, from carbon 1 we go to carbon 2, we have H and OH or HH. OH ribose, only the HH, that's without the oxygen, deoxyribose. So when you are drawing a DNA, that one is HH on carbon 2. On carbon 3 we have HOH if it is a sticky end, and HH if it is a blunt end. And we move to carbon 4. Carbon 4 closes the ring, and carbon 5 hangs outside the ring and connects to the phosphate backbone. So kindly like and share to your friends to also learn something new, and always come back here for more videos like this. And next time we'll be looking at how DNA is replicated or multiplied inside a living organism or outside a living organism. This will be interesting. See you next time.